Morning students, welcome to LEELA'S tutorial. In last class we have discussed about the superovulation process and then how you are going to aspirate the oocytes out and you are going to incubate, right? So here we are going to discuss about in vitro maturation of oocytes. So I said once you aspirate the oocytes, you need to incubate them. Why you are incubating in vitro? In vitro, that is nothing but in lab condition. So here the oocytes which you have aspirated. So here this was the animal reproductive system what we discussed in last class. So in this either by superovulation or by normal process, I said during one estrus cycle the follicle will result in formation of one oocyte, right? So now you can see the oocyte is formed, right? The oocyte, with the help of the scanning probe and the aspiration needle, you are going to collect this oocyte in the collection tube, right? So once you are collecting that oocyte in the collection tube, you need to grow them because these oocytes are immature oocytes. When you fertilize these oocytes with the sperm cells, they will not undergo fertilization, right? So they are allowed to get matured in the lab condition. That process is referred as in vitro maturation of oocytes, right? Maintaining the oocytes with all the sufficient nutrients in the lab condition to undergo the maturation process. So here you can see the immature oocytes are collected from the female animal, then they are grown for 2 days. So why you are allowing them to grow for 2 days? So that the immature oocytes which you have collected, right, they will undergo maturation for this 2 days in lab condition, right? Then the oocytes are allowed to mature outside the body, that is in the lab condition. So one stage of IVF is your maturation of oocytes. See, when you go for the IVF or the in vitro fertilization, it is the superovulation where the hormones are given so that the female animal will produce more number of oocytes. Then the immature oocytes will be aspirated out. Then they are allowed to mature in the normal tissue culture media, and the matured oocytes, see here, these are the stages of maturation. So let us discuss first this how the mature oocytes are formed. So here the single follicle, I said the single follicle will give rise to single oocyte per estrus cycle, right? Per one estrus cycle it will give rise to one oocyte, and these oocytes are immature oocytes, right? So now the immature oocytes, when they are cultured and maintained, right, when they are maintained in the sufficient nutrient media, the tissue culture media, they will grow into the fully grown size or the mature or the maturation starts, right? So here when it increases or fully grown oocyte consists of visible nucleus, right? It consists of visible nucleus. Then for the second day it will start undergoing the meiotic division, right? You can observe the meiotic maturation of the oocyte. So after the meiotic maturation of the oocyte, now after 2 days, now you can see the immature oocyte has developed into mature oocyte. That means the maturation of the oocyte has occurred in the lab condition, that is in vitro condition. So here the mature oocyte consists of the polar body towards the corner, right? So now this mature oocyte is ready for the fertilization process. So now the sperm cells are allowed to fertilize this oocyte so that it can be implanted or it can be put into the uterus of the female animal. So either you are going to, after fertilization, the zygote whatever you obtain, you are going to put it back into the same female. Otherwise, you are going to introduce that into the another female animal, right? If you are collecting the egg from the animal, right, and introducing after fertilization, if you are introducing zygote into the same animal from which you have collected the oocyte, then that animal is called as froster animal or the froster mother. Otherwise, in another case, if you are aspirating the oocyte from this animal, and after fertilization, if you are introducing the zygote into the another animal, right, another female animal, then that animal is called as surrogate mother or the surrogate animal, right? So what is the use of this immature maturation of oocytes? One thing is, we said it is the one of the stage in vitro fertilization process, and another advantage is the female animals with infertility problem, right, where they could not produce any oocytes. The healthy oocytes may not be produced by that female animal. In that case, that can go for the oocyte donor. It can obtain the oocytes from another mother, right, another animal. Otherwise, if there is no ovarian stimulation, right, that means here the infertility problem is mainly because of not forming of oocytes, right? So in that case, they go for the oocyte donor, where they donate the oocytes. Those oocytes will be matured in the lab and they will be allowed for fertilization and then implanted into the female who is having infertility problem. Then that animal can be having the pregnancy and it can give rise to its offspring. Is that clear? So this is the in vitro maturation of oocytes. We'll continue in the next class.